Good afternoon, everyone. My name is Jamie. I'm with the Cape Perpetua Collaborative. Please let me know if you can't hear me or anything, and we'll try to address that as best as we can. Otherwise, let's get going here. I'm going to share a quick little blurb about the collaborative, and then we'll get going with Raquel. Oh, great. Oops. 1 minute, technical difficulties. There we go, right one. Oh, so welcome to our final, this is the last one in our winter speaker series and Wave Makers webinars. Raquel's rounded it out for us with her talk tonight. Welcome to the Cape Perpetua Marine Reserve and Collaborative. We are the largest marine reserve in Oregon, in the state. We span 15 miles from Yachats all the way down to Cedar Head Lighthouse. Here's a beautiful overview shot of our marine reserve, as well as some of our tide pools, some mushroom foraging going on there. We have an array of programs that we're very proud to foster conservation and collaboration within our local communities for scientific exchange, management awareness, and stewardship from the land to the sea in and around the marine reserve. At the bottom of this slide here, you can see an array of partners that we work with all the time. We all just celebrated our 10 years this past November at the Land and Sea Symposium. It's fantastic. So it's only getting better from here on out. Excuse me. Here's a little bit about some of our programs that we offer through the Cape, including our Marine Reserve Ambassador Program, which you can find throughout the spring and summer months at Smelt Sands, as well as our Tide Pool Ambassador Program, which you can catch throughout the spring and summer as well. Marine debris survey monitoring, we do that every month, as well as things like sea star monitoring, shorebird monitoring, and a bunch of different bioblitzes, including our mushroom bioblitz series in the fall. We do currently have a few shirts up for grabs if you would like to visit our bonfire store. They all go help support all of our programs that I just rattled off there. My favorite one happens to be the I like big boffs and I cannot lie shirt. Amazing. So feel free to check those out if you have a wild hair. Otherwise, feel free to connect with us after this. Our website is always up and running. You're welcome to reach out to us at our email, as well as on our Facebook and our new Instagram page as well. So make sure to check us out there. Other than that, let's get this going and meet today's speaker, Raquel Gilliand. Raquel graduated from Stetson University in Florida with her BS in Aquatic and Marine Biology. She spent 2 years interning in the Florida Keys, working on coral restoration projects, and then 2 years in Hawaii, working on more coral restoration projects and wastewater management on the island of Oahu. Currently, she is working on getting her master's degree in marine resource management from Oregon State University. She loves being outdoors, camping, hiking, snowboarding, and surfing in her free time. Heck yeah. All right, so other than that, we will get going here. Raquel, you're welcome to take it away at any point.
For some reason it says that my video is disabled.
Oh, let's see here.
By the host.
There you go.
I can just do share screen if, okay, cool. Hi, everybody.
Hi.
Alrighty, share screen. Oop. Ba-doop. Slideshow. In the beginning. Okay, can everybody see that? Yeah, okay, great. So my name is Raquel, and we're going to be talking about utilizing cooperative fisheries research to better understand harmful algal blooms along the Oregon coast. I'll be first talking about some
Whoops.
Did it cut out?
Yeah, it did cut out.
Okay, no worries.
No biggie.
Let me just try and share again.
There we go.
Okay, can you see that?
Yep.
Okay. Great. Okay, can you see this presentation flow slide?
It's a little slow to load, but it looks like it's trying to. I can see the first slide, but it's not like in presentations.
Okay. Let's see. Got on early. Make sure it was all working. And of course it was working earlier.
It's always how these things happen every time.
Right.
It'd be weird if it didn't.
Yeah.
It looks like it's trying to do something. Oh.
Oh my gosh, I don't know what's happening. Maybe if I just don't do it in big screen, can
That's
you all
fine.
see that?
I think we can all handle that.
Okay, great. Okay, so we're on the second slide where it's background on phytoplankton and HABs, talking about my project, and then we'll go over questions. If you have any as we're going along, just let me know. So secondly, see if this next one loads. What are phytoplankton? So phytoplankton stand Greek for plants, and then phyto also standing for drifters or wanderers in the ocean. There's many different species and types of marine algae, from single-celled organisms called phytoplankton all the way up to multicellular large organisms such as seaweed and kelp. All of these have the common thread of being photosynthesizers, meaning that they utilize sunlight and CO2 to create energy. Phytoplankton are really important because they're the basis for all the marine food webs, and they hold an important place for the health of our oceans, but also human well-being. And we'll talk more about that later. Next, we're going to discuss a little bit about how phytoplankton are super important biosphere engineers and how they provide many ecosystem services to humans. Ecosystem services are defined as any service that the ecosystem provides for us. And importantly, phytoplankton provide half of global oxygen production, and they also do nutrient recycling. And as I mentioned earlier, they're the basis of the marine food web. Next, phytoplankton are also really important for carbon sequestration. As you can see in this graph, phytoplankton take up carbon from the atmosphere, and they either die and or get eaten by zooplankton as either dissolved organic carbon or sinking detritus. This carbon is carried to the bottom of the ocean or carried throughout the marine food web. This is also called carbon burial because the carbon is being buried at the bottom of the ocean, and also allowing that sinking deep water to take that carbon all the way down to the bottom, and then can take up to thousands of years to recycle back to the surface. In this figure from Naselli 2022, we can see about half of the global net primary production a year comes from phytoplankton. And just as a reminder, phytoplankton does primary production, which is the storage of energy through the formation of organic matter from inorganic compounds. So basically photosynthesizers doing their job. Additionally, phytoplankton is responsible for half of the Earth's oxygen production. Phytoplankton represents an important green lung for our planet, and this largely contributes to providing our ecosystems with regulating services, such as air quality maintenance. Now we're going to talk a little bit about the two different kinds of phytoplanktons. There are diatoms and dinoflagellates. As you can see, the white picture and the central picture are diatoms, and diatoms are single-celled eukaryotic algae. Also, so are dinoflagellates, but diatoms are either centric or pinnate, meaning that they're either round or they're long and shaped like that, as you can see in the picture. And diatoms are really important because we can learn about them from studying them and figuring out what they are doing. And then we can learn about the ecosystem and the water quality. Dinoflagellates are also a single-celled eukaryotic algae, and their biflagellate structure allows them to move around or have a little bit more mobility in nature. And these are also the ones that bioluminesce, which is always fun to see if you're on the boat. Next, we're going to talk about the diatom that I work with personally, which is Pseudonychia. There's 58 different species of this organism, and 28 out of those 58 can produce a neurotoxin called domoic acid, which we're going to talk a little bit more about in a minute. And domoic acid is what can cause harmful algal blooms. So what is domoic acid? So both types of phytoplankton that I was speaking about earlier have the ability to produce neurotoxins and create these harmful algal blooms. However, this is a good time to touch on that although phytoplankton provide many of those ecosystem services, such as being a really important ocean lung, they can also create these harmful algal blooms, which can cause devastating effects not only throughout the ecosystem, but also for humans. Shellfish and fish can accumulate domoic acid without apparent ill effects. Research has shown that razor clams accumulate domoic acid in edible tissue, and they're either really slow to expel it, and sometimes this can be up to the order of 6 months before it's completely out of their systems. Additionally, Dungeness crabs, which is one of the largest economic values here in Oregon and the rest of the Pacific Northwest, they can consume these crabs, and then that bioaccumulates in their tissues. And that molecule up there is what the domoic acid looks like. Next, we're going to talk a little bit about the life cycle of what some of these Pseudonychia look like. And it's really important to understand these because it helps us better understand about ocean carbon cycling and how they produce these neurotoxins, and when these harmful algal blooms are occurring, when they're reducing, and when they're finally over. The first stage is going to be our growth stage, which is the cell division. Then we have the reproduction, which is basically gene sharing and swapping. Then quiescence, which is any cell stage that greatly reduces the metabolic rate and ceases cell division, but they also remain viable in this state. And last but not least is cell death. And this is important, like I said, for that carbon cycling, because when the cells die, they sink to the bottom of the ocean. And then this is also important for bloom terminations. When a harmful algal bloom is over, that's when we get all the cell death. And harmful algal blooms cause negative impacts on ecosystems, as I was touching on earlier, and throughout the production of natural biotoxins by certain kinds of microscopic algae. So they cannot get stuck in fish gills and can discolor the water, causing cloudiness and not allowing sunlight to pass through the rest of the marine ecosystem correctly. Marine biotoxins are normally present in the amounts that are too small to be harmful. However, molluscan shellfish, which are like the hinged shells, so like bivalves, clams, oysters, they can all take up filter feed these phytoplankton, and then it bioaccumulates in their tissues, and then the crabs can eat these clams, and then that gets caught up in their tissues, and then we eat them. This is some imagery of Western Lake Erie during a harmful algal bloom in September 26 in 2017. This scum was shown here down in Toledo that stretched all the way to Lake Ontario. This photo was taken by NASA satellites, and there's three main factors that make phytoplankton bloom or grow exponentially quickly. Number one is nutrients, number two is sunlight, and number three is warm water temperatures. In no particular order, it's a combination of all three of these factors coming together. Next, growth varies seasonally due to the number of sunlight hours and sunlight intensity throughout the globe. In high latitudes, there's more blooms in the spring and summer. Conversely, phytoplankton populations decrease in the summer in the subtropical oceans. There's warm surface waters and less dense cold waters that are found deeper in the ocean. And when we have large storm events, as you can see here, this wind goes down, pushing the water offshore, and then through Ackermann transport, and then allowing upwelling of that cold, nutrient-rich water, which phytoplankton love. So all of this that we talked about so far is affected by climate change. These ecosystem services are constantly being changed because of climate change and all this bad effects that are happening to our world. So a little bit about what we're doing about it is where my project comes in. My personal project is funded by NANUS and Oregon State University, but there's also a bunch of other organizations and projects that are working on harmful algal bloom monitoring here in the Pacific Northwest and throughout the world, such as the Northwest Fisheries Science Center, the harmful algal bloom team over at NOAA, and the Oregon Department of Agriculture and ODF&W. Some overarching goals of my research are to co-create a regular monitoring program for HABs by collaborating with local fishermen to collect phytoplankton samples, and to equip the regional NANUS HAB monitoring system with offshore water samples. In this picture here, I am on the RV Alaka, which is an Oregon State University research vessel, and I am collecting water samples offshore, and we use these sampling kits, which are equipped with small vials to collect water samples and temperature gauges, and then a data sheet. Some primary questions that I was hoping to answer with this research is to better understand, are Pseudonychia always present from June to November off of the coast of Oregon? And if so, in what abundance? Can we detect Pseudonychia offshore before it turns into a large bloom? Can we illuminate some of the drivers of Pseudonychia? What are the benefits of cooperative fisheries research, working with fishermen day in and day out to better understand these harmful algal blooms? What roles does all of this play in fish enclosures, timing, and placement? What is the relative abundance of Pseudonychia off the Oregon coast during the summer months? And what are the relative abundances? Are they higher than normal? And what environmental data correlates with spikes in Pseudonychia? Are we looking at salinity, sea surface temperature, winds, seasonal upwelling? There's a lot of research that's already gone into this, but as I've stated before, we need to figure out more research to be able to better understand it. It's really important to ask these questions because the more we understand about this, the more we can better predict and hopefully help coastal communities that are being negatively affected by these harmful algal blooms. What have some of the methodologies looked like so far? I've recruited fishermen from commercial fishermen all the way to charter fishermen, and they've collected water surface samples for me, water temperature and GPS sites. And then the fishermen take out these kits regularly and they get swapped out. I pick up these samples and I run them through this crazy looking machine, which is called a flow cytometer. And it runs the water samples through a camera and a laser so we can take pictures of the actual phytoplankton. And as you can see here in this slide, this gray box over here is what some of the pictures look like of the phytoplankton samples that we get. And so far we've recruited 7 fishermen and 3 scientific vessels, collected 271 water samples, all up and down the Oregon coast. We've learned a lot about water surface temperatures, and this graph right here in red are samples from June to September of the sea surface temperature. And we've collected salinity, and we've also run all of these through the IFCB, which is a long process. And since I'm talking to you all from the Cape Perpetua Reserve, I just wanted to point out some of the samples that we've collected near the Cape Perpetua Reserve. So as you can see here, Cape Perpetua, this purple dot is the closest one north of it. And then all of these other little orange dots are other samples that the fishermen have collected water from, which I think is really cool. And then just a little up close example of one of the samples, which had a large abundance of Pseudonychia on this specific slide. And all of these are Pseudonychia. And then this is what one of those pictures looks like up close. And as you can see, the scale bar here is 10 microns. So that's really, really, really small. Definitely impossible to see with just the human eye. So some next steps are to manually classify phytoplankton in these samples. So I have to now go through and click each one and say, this is how many Pseudonychia are in it. This is how many diatoms. This is how many nanoflagellates and so forth. Then I'm going to classify them into thick versus thin. This helps us better understand which ones potentially are harmful and might be producing domoic acid and which ones might not be, because the thick do usually produce domoic acid, whereas the thin usually do not. Again, there's exceptions to every rule in science. And then we're going to overlay these abundance results with spatial mapping that I just showed you. And then we'll overlay this with sea surface temperature and salinity, and we'll try and get a better picture of what's going on overall. And we're also continuing this into the summer, May through October. So I'm currently recruiting for commercial and charter fishers all up and down the Oregon coast to collect water samples. We're looking for about 16 bottles per week up to. So if you can bring in 1 bottle a week, that's great. If you can do 16 bottles a week, great. Really flexible with what works for you. And each sampling takes about 5 to 10 minutes. And it's also okay to run the sample out of a deck hose, which is really important for the charter fishermen. So you don't have to be throttling down the boat. And if you're interested in getting involved in this project, please get in contact with me. There's my school email and my personal cell phone on there. And now, if you have any questions, I'd love to hear from you. And thank you so much for your time. And again, this is my contact information. And then there's references here too, if you have any questions about those. All right, so it seems like people can get back on video. It says the host needs to let them on. Did I maybe become the host by accident? Let's see.
Hey, I cannot, for whatever reason, get my video to come back on. It says the host needs to allow it. But that was a fantastic presentation. Either way, it doesn't matter.
Thank you so much.
Heck yeah, that's great. Yeah, does anybody have any questions or comments or anything like that? It looks like Linda Collins is saying, looking forward to hearing the results. When do you think you might have any kind of quantifiable results or data?
Yeah, so I am going to be finishing my master's thesis by September. So we will have results by then.
Ooh, exciting. You'll have to let us all know how it went.
Yeah, definitely. I'd love to. And yeah, keep your eyes out. And I'll be publishing in at least the OSU student publication, if not another one, hopefully.
Awesome. Well, great.
And I can't see the chat, so I don't know if
It's okay. Joshua just asked, what does Raquel want to do after she graduates?
I will be hopefully getting some kind of job with either NOAA or ODF&W. I'm really looking forward to being a marine resource management and taking all of this amazing field work, real work, and classes that I've learned and applying it.
Yeah. Anne says, thank you so much for all of your work in this interesting field. Truly, like, thank you. It's one of those things that I don't think maybe even many folks know to have on their radar to think about. So it's really cool having a glimpse into this. So yeah, we thank you so much. Does anyone have any other questions or comments? I don't think so. So other than that, make sure to reach out to Raquel if you do have anything or want to know how her research is going down the line come September.
Or if you want to get involved as well.
Yes, heck yeah. If you would like to get involved, that'd be really neat. Make sure to let her know as well. Other than that, Raquel, thank you so much.
Cool. Thank you.
You betcha. Have an awesome rest of your night. Everybody too. Have a great rest of your night.
Bye.
Bye.